So I'm Joe Moritz. I've been a faculty member at West Virginia University for the past 22 years, and I've got a three-way split appointment in research, teaching, and extension. And a large part of my research appointment is associated with feed manufacture research. So I want to give you a little bit of background on why soybean meal processing is important to someone that does feed manufacture research. So when we perform feed manufacture research, typically we change variables at the mill, and that in turn has an effect on two primary factors, pellet quality and nutrient availability, which in turn affect meat bird performance. In order to really understand what's going on with either pellet quality or nutrient availability, we need to hold one of those factors constant. So for example, if I'm going to hold nutrient availability constant, I would make a large volume of pelleted feed, and then I would grind a portion of those pellets back through my roller mill, and then I could mix pellets and ground pellets together to look at different percentages of complete pellets when I feed birds. And when I do that, my results are fairly consistent over the years. So here's a series of studies looking at increases in whole pellets presented to birds and the effect on feed conversion ratio. So you can see we move from 40% pellets to 70% pellets, we get a 5-point reduction. 50 to 70, a 3-point reduction, and 30 to 80, a 6-point reduction. However, when we try to hold pellet quality constant and instead focus on nutrient availability, the results are less consistent. So a made-up scenario here would be if I would take formulations and I would condition them at, let's say, 70 degrees C, 80 degrees C, and 90 degrees C, then I would take all those resultant pellets, run them back through my roller mill to eliminate feed form, and then feed the birds. So I'm just looking at what's going on with the thermal process of feed manufacture and the effect on birds. So when I do this, sometimes increased thermal processing decreases performance, but sometimes increased thermal processing increases performance. When it decreases performance, it's sometimes easy to explain. I can say that, well, amino acid digestibility was decreased, especially for more heat labile amino acids, or perhaps enzyme activity was decreased if I'm adding mixer-added enzymes. But when increased thermal processing increases performance, we have a more difficult time explaining that. In the past, we've speculated that in some cases, the Aluron cell lysis occurs through the corn that was in the diet formulation. In other times, we speculate that the trypsin inhibitor was decreased because the formulation was utilizing an underprocessed soybean meal. So if I look back through the literature, there's a recommendation that trypsin inhibitor at about 7.6 TIU per milligram is the recommended threshold. So activities at that level or above would be detrimental to performance. However, other papers go down as far as 1.5 TIU per milligram, and they show detriment to performance. So when I see these results in my lab and we happen to retain some soybean meal samples and send them out for analysis, oftentimes we're seeing levels that are in between those two. So this helped me to generate a couple research questions. One, does feeding underprocessed soybean meal decrease performance and amino acid digestibility of modern day broiler chicks? And I'm not talking about extremely underprocessed meal, something that's in the more attainable range. And then if that is true, can standard conditioning and pelleting practices mitigate nutritional issues with underprocessed soybean meal? So in preparation for these experiments, I learned quite a bit, and actually you learned the same thing today in the symposium, so that was very nice. As Hari and Lisa had mentioned, soybean meal contains several heat labile anti-nutritional factors. So it's more than just trypsin and chymotrypsin, but we've got lectins, and there's even a Brazilian group that's looked at soya toxins and soybean toxin and even urease per se as being detrimental to rodent models. And as Gordon had mentioned, soybean crush plants vary. So these plants are going to all use a toasting step, but the retention time and the temperature during that toasting step can vary. And also other factors such as the number, size, and design of decks associated with the DTDC unit can change. And then, of course, we've got variations in cultivars. So these factors make it difficult to determine a set optimal temperature and time combination for plants. And then finally, as Dr. Parsons mentioned, it seems that these metrics are somewhat fluid. There's some different recommendations. When we talk about the original urease pH change recommendation and how that's evolved to maybe it's just useful for under-processed soybean meal. And if we're going to talk about over-processed soybean meal, we should really talk about solubility in potassium hydroxide or solubility in water. And then I also mentioned the variation that we see for trypsin inhibitor. So this led us to 3 objectives. The first objective was to obtain 3 soybean samples from a commercial solvent-extracted crush plant, and those samples were called under-processed, peak-processed, and over-processed soybean meal. And then we characterized these meals using the trypsin inhibitor activity assay, both the AOCS as well as Hari's USDA ARS methodology, and then the standard methods, urease pH change, potassium hydroxide, and protein dispersibility index. We then took those samples, formulated them into mash diets, and fed them to chicks in raised wire cages and looked at performance. And then finally, we took the under-processed diet and the peak-processed diet, formulated them into diets, and then conditioned them at either 70, 80, 90 degrees Celsius. Again, ran all those pellets back through a roller mill, eliminated feed form, and we fed them to chicks in raised wire cages and looked at performance as well as apparent ileal amino acid digestibility. So a commercial soybean plant was visited in 2023. This plant at the time was processing new crop beans, and they described 3 different samples that we obtained, under, peak, and over processed. The way they obtained these different samples was they varied the rate of beans going into the DTDC unit. And as you can see in my picture, we're not talking about extremes in processing. We're not talking about white flakes compared to something that would be really dark brown. So these are more subtle variations. And I was happy for that because I believe this is what I've encountered in the past in my lab. So as I said, they varied the rate into the DTDC unit. So beans conveyed at 100% as per manufacturer recommendations for equipment, had about a 26-minute retention time in the DTDC unit, and we called that peak processing. Beans were conveyed at a faster rate, and since the DTDC unit runs on volume, this decreased retention time, which we would believe would be under 26 minutes, and then beans were conveyed at a slower rate into this unit, which should increase retention time, and that's how we came up with the under and the over processed. Also, data shared from the plant showed that all of these samples hit about 109 degrees Celsius meal temperature at the sparge deck. So I believe that really the difference was retention time in this unit. The characterization of the meals was associated with trypsin inhibitor with both the AOCS method as well as Hari's new method, urease pH rise, and then solubility in potassium hydroxide and solubility in water. Methodology for our mash feeding included day-old male Hubbard by Ross chicks, 12 replications per treatment, corn soybean-based diets, birds were in raised wire cages, and we focused on performance. And then on our final objective, we took the 2 diets that we conditioned at 70, 80, or 90 degrees C, all for 30 seconds. And then we ran that through a 32 by 4.5 millimeter die. That's 1.25 inch effective thickness by about 3/16. Once we formed those pellets, we ran it back through a roller mill, eliminated feed form, and then fed birds in raised wire cages. We looked at performance as well as apparent ileal amino acid digestibility on day 18. This slide just kind of puts together the materials and methods with the objectives. So just for clarity, again, we characterized 3 soybean meal samples, under, peak, and over. We then formulated diets with the under, peak, and over at a decreased plane of nutrition to better see what the differences of those meals would be. So we ran about 85% crude protein and 85% digestible amino acid requirement. We threw a 4th diet in there where we ran our peak process diet at 100% crude protein and 100% digestible amino acids as a positive control. And then our final objective, we looked at 2 formulations, the under and the peak, pelleted those using conditioning of 70, 80, or 90, reground, and again, those diets were on a lower plane of nutrition. We looked at performance and amino acid digestibility. So here is the proximate analysis data obtained from the samples, and we have data both from NIRS, from the plant, as well as AOAC analyses. that we sent out from WVU. You can see that crude protein increased and crude fat decreased as we increased retention time in the DTDC, and that may have had to do with the conveyance into the DTDC. You also notice that the crude fat levels are high, and that's because this plant was making degummed oil at the time. So as Gordon had mentioned, the lecithins were added back to the meal. Here we're looking at the characterization, and we had 4 labs. Each lab was sent 10 samples, so we're able to get some data and some understanding of within and across lab variation. Some labs performed all the analyses, others just did a few. I also have on the right-hand side kind of a cheat sheet of some of the standards that you can compare that back to. So I can walk you through here. If we look at lab number 1 for urease, you see we get a pH change of 1.25 for the under. We move down to peak, we get a pH change of 0.29, and we look at overprocessed, we get 0.09. So we see a nice decrease in pH change as we increased retention time in the DTDC. However, I'll also show you, or let you know, that both lab 1 for peak and under would be above that 0.2 threshold, so it would indicate that both of these were underprocessed. Lab number 2 instead shows a drop for underprocessed from 0.07 to 0.04 between under and peak, and then a further drop at 0.03 for the overprocessed. And in this scenario, all of these would be adequately processed. And then lab number 3 for urease was not able to differentiate anything, any differences between these samples. Protein dispersibility index, potassium hydroxide, they were just about within spec as would be suggested for optimally processed meal. And if we look at the trypsin inhibitor assay, you can see that both lab 2 and lab 4 had very similar levels for the underprocessed and the peak, but both levels dropped when we're looking at the overprocessed meal. I also analyzed the complete diets. I thought there would be value in doing that. So this is data that's associated with just one lab, but that one lab was sent 10 samples. Here we also see that trend for urease. Even though they're complete diets, you can see that drop in pH change moving from the under to the over. And then the other assays were not in line with what was done at the plant. Okay, now let's look at what the birds told us. On the left-hand side of the screen, I have live weight gain. On the right-hand side, I've got feed conversion ratio, and this was a 0 to 21-day study with MASH feed. You can see the positive control had a high live weight gain and a very low feed conversion ratio, and this, of course, was by design. When we look at our diets with a lower plane of nutrition, you can see the diets containing underprocessed soybean meal had a very low live weight gain and high feed conversion ratio, and then diets containing either the peak or overprocessed were intermediate. So I believe that these data would suggest that, yes, there was a problem with the underprocessed soybean meal, likely associated with anti-nutritional factors. Okay, moving on to our third objective, we took the underprocessed and the peak processed diets, or samples rather, formulated them into diets, and then conditioned at 70, 80, or 90 degrees Celsius. If we focus just on the underprocessed rows there, you can see that for urease, we again see a drop moving from under to peak, 0.029 to 0.008. And we also see a drop in protein dispersibility index from 22.03 to 20.76. So these are somewhat in line with what happened at the crush plant. But when we move into differences among the underprocessed sample to 70, 80, and 90C, we lose those trends, except for protein dispersibility index when we focus on the underprocessed, and you can see how that continually drops, 22.03, 19.85, 19.83, and then 14.73. So maybe there's some value there. So what the birds told us in this case, again, we're looking at live weight gain on the left, feed conversion ratio on the right, and we're looking at interactions here. And it's an interaction between diet, including the particular soybean meal type, and then conditioning temperature. So for live weight gain, we can see that diets containing underprocessed bean meal increased live weight gain as conditioning temperature was increased. And the opposite occurred for diets that contained the peak processed meal, where we see a drop in gain as conditioning temperature increases. I want to point out that if we look at the 70 degrees Celsius conditioning temperature for the underprocessed meal diets, that gave us our lowest gain. And then we see for the peak processed diet at 70°C, we get our highest gain. But then just taking that underprocessed diet and conditioning it at 80°C restored live weight gain to that of the rest of the treatments. For feed conversion ratio, we can see that as we take the peak processed diet and increase conditioning temperature, we see an increase in feed conversion ratio. And then there was no difference for the underprocessed diet across increasing conditioning temperature. So we also looked at apparent ileal amino acid digestibility, and this was again on day 18. And here we're looking at lysine. It's also a significant interaction. And we had the same significant interaction for all amino acids that we tested. So we're seeing that for the underprocessed diet, as we move from 70 degrees Celsius to 80 degrees Celsius conditioning, we're increasing amino acid digestibility, and there did not seem to be an effect for diets containing the peak processed soybean meal. So in conclusion, for the characterization and MASH study, current analytics for anti-nutritional factors of soybean meal, particularly the potassium hydroxide, protein dispersibility index, and trypsin inhibitor assay, may not have been sensitive enough to detect small variations in soybean meal processing, and I believe that's what we were looking at, small variations. Underprocessing soybean meal did in fact result in a reduction in live bird performance when included in a MASH diet, and then pH change due to the urease assay from 2 of the 3 laboratories best indicated crush plant processing changes, and it aligned best with live bird performance. When we look at conclusions from characterization of the complete diets and the ground pellet study, current analytics for anti-nutritional factors found in the complete diets may not be sensitive enough to detect small variations in the processing of soybean meal. Conditioning the underprocessed soybean meal diet above 70°C increased amino acid digestibility and restored live weight gain to levels similar to peak processed diet, mitigating potential anti-nutritional effects. In conditioning diets containing peak processed soybean meal at temperatures exceeding 70°C decreased live bird performance, but did not affect amino acid digestibility, at least at that day 18 time point. So my take-home message is that underprocessed soybean meal, at least the underprocessed soybean meal that we obtained in this study, can decrease amino acid digestibility and broiler chick performance, presumably due to anti-nutritional factors. However, conditioning diets containing underprocessed soybean meal at 80°C for 30 seconds restored amino acid digestibility and performance, and that should be good news to any pellet manufacturer of meat birds in the United States, because that's fairly attainable. Thank you so much for your time. I want to thank USB for financial contributions. I want to thank my graduate students. The young lady holding the fish there, she's going to defend her PhD on Friday, so I appreciate her, and if you're looking for new talent, let me know. Thank you.